In this series of videos, we're going to talk about the nitrogen cycle and different aspects of how nitrogen is transformed on biological pathways and how it's ultimately assimilated into biomolecules. So first, we're going to start with a brief introduction to the nitrogen cycle. So as you know, nitrogen is important because it is incorporated into a number of biomolecules, including nucleotide bases to form DNA and RNA, and also in amino acids. Now, the largest source of nitrogen on Earth is dinitrogen gas, or N2, from the atmosphere. However, the problem with N2 is that it is kinetically inert and not very bioavailable. So the first thing that needs to happen in order to become bioavailable is something needs to convert it from this very kinetically inert form into something easier to use by life. And so what I'll be describing here is a series of steps on the so-called biogeochemical nitrogen cycle. So a number of, for instance, there are a number of different pathways towards formations of bioavailable nitrogen species. However, I'm going to focus on, for the purposes of this, the biological pathways, which means these are all mediated by bacteria, archaea, and even some fungi. So in the first step, which we call nitrogen fixation, dinitrogen gas from the atmosphere is converted to ammonia or ammonium. And so this is the common form of what we think of as fixed nitrogen. And then the next step in this core nitrogen cycle is that ammonia or ammonium is oxidized to oxidized nitrogen species such as nitrite or nitrate. Okay, so these are still usable fixed or bioavailable and fixed forms of nitrogen. Now we have to be able to close the loop. So in the modern nitrogen cycle, nitrate is finally reduced back to dinitrogen on a pathway called denitrification. So how do these species ultimately end up inside biomolecules? So these happens through what are called assimilatory pathways, which can take either ammonia, ammonium, or nitrate and incorporate those into amino acids and nucleotides by various biosynthesis pathways. So if we look a little bit closer, there's several sources for ammonia biologically. And so first, in addition to the fixation pathway, ammonia can also be reclaimed from organic nitrogen sources. Again, that can be from, for instance, nucleotides or amino acids that have been generated by decomposers on pathways called ammonification. And nitrate is actually first converted to ammonia or ammonium by assimilatory nitrate reductases. And so after reduction of nitrate to ammonia, this then goes through similar assimilation pathways. And so the reservoir for nitrogen, for instance, in humans, isn't typically ammonia or ammonium because it's toxic to us. And this reservoir instead is held in glutamine, where the amino acid glutamine, where it is held until it is needed for biosynthesis. So to show this, we're going to look at the activity and mechanism for the enzyme glutamine synthetase. All right? And so this is actually stored by starting with the amino acid glutamate, and then in an ATP-dependent process, ATP is hydrolyzed and form a reactive intermediate, which is a glutamal phosphate. Ammonium reacts with this to form glutamine, and then this becomes a nitrogen reservoir for later biosynthesis. One example of this is glutamate synthesis itself. So for the enzyme glutamate synthase, glutamine, the ammonium is released from glutamine, converting the amino acid back to glutamate, and then forming ammonia or ammonium. This is then incorporated into alpha-ketoglutarate, which, as you remember, is an intermediate of the Krebs cycle. So the enzyme reacts the ammonia with alpha-ketoglutarate to form the amine, so alpha-aminoglutarate, and then electrons that are ultimately sourced from NADPH are transferred through electron transfer pathway through flavonoadenine dinucleotide and flavomononucleotide to reduce the aminoglutarate to form glutamate as the product. And now, and so that is how glutamine, for instance, can be used as a nitrogen source in biosynthesis. So that's one example. And so, as you see, both of the assimilatory pathways actually start from ammonia or ammonium, and that is what goes into these assimilatory pathways into glutamine. So ammonium is the key nitrogen source that we'll be discussing in the later videos.